Dr. Begley.
Good afternoon.
Irreproducibility is clearly a community problem that requires a community solution. But scientists are under considerable pressure, as I already mentioned, the pressure to publish, to obtain grants, increasing competition for dwindling resources, a growing focus on translational research, and an increasing complexity of science and technologies, just to name a few. How do we design solutions, incentivize the community, to make the development and adoption of standards a priority in the face of these significant challenges that every scientist is facing? It's all yours.
Thank you. Well, I wish I had a short answer. I think this is actually going to take quite a lot of work, and I think we need both a carrot and a stick approach. I think we need to offer incentives. Excuse me. We need to have incentives that encourage people to do what they really want to do, and we've also must have procedures in place to discourage behaviours that are not appropriate in terms of advancing science. The position that I come from is fundamentally the vast majority of people that are involved in this exercise want to do the right thing. They want to discover cures for cancer. They want to know how to improve human health. They also face considerable practical challenges from day to day, and part of our role is to try and make their life a little bit easier from that point of view. I think what we have here is a shared responsibility. It's a responsibility that's shared principally between, in my view, three groups: the investigator and their institution. I think they principally must take responsibility for the data that is generated and the way that it's presented and reported. I think the funding agencies are also critically important, and I think, in truth, they have the greatest opportunity to modulate behaviours in this area. Finally, and perhaps least important, I think, are the journal editors and the reviewers. I think they are important, and they've already taken steps to try and improve and address this issue. But I think, honestly, that they are probably the least important of those three. And in my view, the principal responsibility rests with the investigators and their institutions. I'll come back to each of those in a moment. I'd note that I think the issue was actually solved by the clinical community in terms of reproducibility of data and so on several decades ago. So when I was a young, it's hard to believe, it just seemed like yesterday, a young clinician, the sorts of things that were acceptable then in the 1970s in terms of clinical trials and so on are totally unacceptable today. So today we understand issues of clinical trial blinding, randomisation, pre-specified hypotheses, data exclusion, statistical analysis, and so on, and that's built into our system. We've accomplished that over the last several decades. We don't have really anything comparable to that in the preclinical arena. So it is something that can be done. It's something that I believe would significantly improve the quality of our preclinical research. Getting back to those three areas that I mentioned earlier, taking what I think is the least important, the journals, the editors and the reviewers, they clearly have an important role to play, and I think over recent months they're to be congratulated on taking that role seriously. There have been many editorials addressing this problem in the top tier journals, and a number of the journals have rewritten their guidelines to authors, recognising that this needs to be more rigorous. I think they do have an important role to play, and I'm not trying to trivialise it, but at the same time, they can only do so much. Because if I, as an investigator, submit a paper where I've left out, for whatever reason, 3 of the studies that I performed because I liked this one best, a reviewer, an editor, cannot know that that information was highly selected in that way. Again, getting back to the clinical arena, that would be totally unacceptable. But it's honestly standard practice in many of the preclinical publications. I think the funding agencies really have the most opportunity to influence our behaviour here. They can put in place mechanisms by which results that are reproducible and reliable are rewarded, and investigators who fail to comply with those sorts of standards are not. And a number of funding agencies are looking at that sort of process, and I think that that will benefit all of us. Finally, the investigators and their institutions, I think some investigators are able to self-regulate, some are able to self-censor. There are many, however, that are not, and they are too keen to get papers in high-quality journals because that drives grants, fame, the next generation of research. And in that situation, I think the institutions have a much greater responsibility to play than what we've seen them willing to accept so far. So the institutions generally are prepared to bask in the kudos, the fame, the fortune that their investigators generate as a result of publications in the top-tier journals. But generally, the institutions are seldom willing to rein in those same investigators when they take liberties with their research or not completely disclosing all of the data as it should be disclosed. So my conclusion is that we need both a carrot and a stick. We need both incentives and disincentives. As a society, we get what we incentivise. If we put the incentives in place, I'm sure we can modulate behaviour. I think the principal responsibility rests with the investigator and their institution. The funding agencies have a very important role to play. And although we quickly turn to the journals and the editors, I think that their role is less important. And again, I would observe that we've already solved this problem in the clinical arena several decades ago. So I believe that we should be able to solve this same problem again in the preclinical space. Thank you.
Thank you, Glenn. I also want to mention that those who are viewing this online, please email your questions to ask@gbsi.org. And after the panel, we'll be reading those questions. Mary Lou Gantzer, good afternoon.
Good afternoon.
The greater use of standards may not necessarily reduce costs. In fact, their implementation may cost more in time and money up front, but the long-term cost savings and quality improvements may be significant. And actually, this situation is similar to the case for companion diagnostics with targeted therapies, increased cost up front for a long-term cost savings and improved patient outcomes. How do we address this challenge in our efforts to promote the increased use of standards in life science research?
I think that's an important question that everybody is looking to find an answer to. Certainly the perception that exists is that the development of standards, adherence to standards, can be incredibly expensive. But I think if you take a step back and really carefully look at that situation, what you discover is that while there may be some upfront costs, they're minimal compared to what the payback is in adherence to those standards. Probably the best example of that is the ISO 9000 standards that those of us who work in industry have followed for many years. Those are voluntary standards. But if you don't follow them, chances are you're not going to be successful doing business throughout the world because people have come to expect that their vendors will follow ISO 9000. That whole concept has extended beyond industry into the clinical laboratories. Quality systems have become very much a focus in clinical laboratories, and that is one of the things that CLSI is engaged in. It's helping labs worldwide understand how to implement clinical systems, clinical quality systems, so that they can help guarantee the quality and accuracy of the work that they do. So certainly one takes a step back and says, well, there's expense involved in developing standards. Yes, there is. There's no question about that. Whether they're material standards or the consensus-based document standards, there definitely are costs associated with those. The material-based standards may be quite expensive depending on how many iterations an organization needs to go through before they develop something that truly will function as a material standard, because believe me, they don't all do what you want them to do. And so you take several passes at that. For the consensus-based document standards, where you're bringing together experts in the field to discuss and document best practices, those are not nearly as expensive. Our best guess, based on the standards that we have from CLSI, is that it costs us about $10,000 to $12,000 per document that we develop. And that cuts across the entire clinical laboratory community. And we generally have about 200 standards that exist that are used throughout the world by both industry, government, and those in the healthcare professions. So why do they do it? They do it because in the long run, they're going to get the quality they want. There's nothing worse than generating a result that a patient is going to be treated based on in one laboratory and then finding out that, whoops, there was something wrong with the assay. The results I got weren't right. The patient treatment was not right. That results in harm to patients, which is what people throughout the healthcare community are trying to avoid at all costs. So standardizing the way things are done, identifying those little details that are the things, the pre-analytical is what is usually the issue. Identifying all those things and having standards in place that are routinely followed can help eliminate those kinds of things from occurring. And I can share with you one of the stories I have heard in my work outside the U.S. that is one of the saddest stories I ever heard, and that involved a young military person from the U.S. who was working outside of the U.S. in one of the developing countries helping build infrastructure, wound up contracting an illness. The initial tests that were done in country were incorrect. We don't know if that was because a poor quality assay was used, the assays may have been adulterated, or the laboratory may have just performed them incorrectly. But the initial results were incorrect. So as the individual became sicker and sicker, they were hospitalized. Unfortunately, the next step that happened was the drugs that were used to treat that individual had been adulterated, either by the distributor of those drugs or by the institution. Again, we don't know which. But as they continued to go ill, they were actually airlifted. In this case, they were airlifted to Germany, where they died of malaria, something that just should not happen in today's world, although there are a number of people who continue to die of malaria every year because of things like this. So getting to the point where all of these activities are highly standardized comes with a little bit of cost up front, but the downstream recovery, economic recovery, is just incredible. So upfront costs are there, but minimal in comparison to the gains in positive outcomes that are achieved. So I think all of us on the panel agree that people in the research community don't knowingly try to fudge their data or produce results that can't be reproduced by anybody else. It's probably more a lack of knowledge or understanding of how to do things in the best quality way that is the impact. So I encourage us to take a step back and not think about any of those upfront costs, but really to think about what the overall benefit is going to be in whatever area of life sciences research we're involved in, whether it be therapeutics, treatment of any kind, or the diagnostic piece. The payback is so enormous that to do it right is just the most wonderful thing in the world.
Thank you, Mary Lou. And in fact, some of the things you alluded to, perhaps it'll come up as a question in a few minutes, but it does beg the question that I've thought a lot about myself, which is why specifically in basic biological research is there such a dearth of standards as opposed to in the clinical world, or even in the world of cell phones or computers, where they simply would not have succeeded in producing a workable product without standards.
And I think one of the differences is if you make a mistake in an area where you're actually impacting somebody's health and life, it's much more visible than if you have a cell phone that doesn't quite do what it's supposed to do. And in life sciences research, sometimes it's so far down the road from the researcher that you don't really understand the implication if you come up with results that are really irreproducible.
Thank you. Yvonne, Reid, this is a bit of a loaded question, but if there was one standard, either materials or process, that if implemented overnight would have the greatest impact on irreproducibility, what would that be, in your opinion?
Okay, so I'm going to answer this question as a case study of a recent standard that was issued through ATCC Standard Development Organization. In 2009, some concerned scientists came to the ATCC Standard Developer Organization, and they wanted to establish a consensus standard on the authentication of human cell lines using STR analysis. And this international working group, which was made up of individuals from biotech, pharma, from industry, government, cell banks, wanted a standard that was very easy to use, that was inexpensive, which was consistent and robust. And so in January 2012, the ANSI, the National Standard Institute, issued the standard on the authentication of human cell lines using STR analysis. It is estimated that 15% to 20%, and in some cases, some published data show 30% of all human cell lines being used, or all cell lines being used in research, are misidentified. This misuse has caused irreproducible results, loss of time, millions of dollars wasted, and has also led to false data. And this can be easily resolved by just using STR analysis to uniquely identify human cell line. You can actually tell one cell line from another if you were to implement this procedure right away. And what we have found is that since the implementation of this standard, there have been some very positive outcomes. So one standard can actually make a difference. Journals are now requesting that authentication be prerequisite for publication. NIH, as a funding agency, is recommending cell line authentication. Journals have begun to publish retractions. Actually, Nature was one of the first to have done this. In 9 years of publication, they actually published their first retraction on the misuse of a cell line that was misidentified. Testing laboratories are actually now using these, the standard as standard protocol for operating and for the identification of human cell lines. We have now seen scientific professional journal societies who are at their annual meetings, who are having symposia and webinars that are dealing with best practices in tissue culture as a way to mitigate or to combat the use of misidentified cell lines. Several members of our working groups have written peer-reviewed articles and also have been asked to present, to make presentations at scientific meetings and at webinars in an attempt to bring awareness about the misuse of identified cell lines. There's also now a list of 400 or about 400 misidentified cell lines that have been made available to the scientific community. We have worked with NCBI to actually put together a public database on all the STR profiles on human cell lines. So any researcher who is wanting to see what their profile should be, they can actually look at the biosample database in NCBI. They can also upload their material into this database to be able to, for that to be reviewed by others. So just one standard actually can make a difference. And we have shown that within it's less than 2 years since this standard was published, that it has had a great impact.
Thank you, Yvonne. I was remiss and wanted to know if the panelists had any comments on Yvonne's response or the other questions as well that we've already been through. Okay. Last but not least, Professor William Bentley. This is a very similar question. If there was one policy, if implemented overnight, would have the greatest impact on reproducibility, what do you think that would be?
Well, thanks. So actually, maybe I'll respond to some of the other comments in my answer to this question.
Terrific.
Being the sole academician, I think I have a responsibility here also, because I noticed a little bit of a poignant kind of point to the academic community. That having been said, I'm definitely glad to be here and be part of the solution as opposed to being part of a problem. I think I should put into context a little bit the word reproducibility in your question, maybe answer that as it's more of a continuum, I think. And the motivation for the question is more about the economic pressures that we face right now and the loss and profit to the national investment in research. And trying to make sure that the investment that we have is translated into products, and especially in the life sciences, to bettering human health. So that actually also brings on a difference between what is a standard, what is a validated method, and what are reproducible experiments. And so really there's a continuum there, and folks in academia as well as in various research institutes deal with all of those. There is a fundamental question, a fundamental difference between the academic researcher who responds to the reward structure, whatever it might be, and someone in a clinical environment and someone in a company. The academic researcher in the university, the goal is to discover and create new knowledge. And by definition, that cannot be standardized. And so you have to have a recognition that the motivation for an R01 or for a 6-1 if you're in the DoD world, or for an NSF grant is different. And the desire is to create new knowledge. That having been said, there are certainly pressures, and a lot of people, I'm an engineer, engineers think about solving problems and translating innovations into products that actually might come to a realized solution. We have to recognize that whole sort of continuum. And so to sort of answer the question about if there was one policy, might suggest that there might be a way in which the publishers, the research sponsors could ensure that translation is part of the research in the first place. So maybe there is an add-on to make sure that if there is a standard, or if you as a PI could develop a standard out of something that you're doing all the time, then here's an extra bit of money that you might be able to use towards that. This could be enabled by NCATS, or like we have the FDA, the Center of Regulatory Science and Innovation. I-Core in NSF is a program set up essentially to help translate innovations into practice. And so another way to address this might be that if a researcher was encouraged, a policy might be in place where the requirement would be that if there is a standard for a particular assay or a particular cell line and so on, you have to use that. The academic community responds to the reward structure. And so if the funding agencies, and I also think the journals have a little bit more of a role to play, because there's increased pressure to take a journal article that I would have written 20 years ago that was 20 pages to make it 5 paragraphs. And you can't include a lot of content in the 5 paragraphs. On the other hand, journals do have an opportunity to submit supplemental materials that's on the website, et cetera. And that could be a repository so that every investigator across the country could make sure that if I was doing my experiment in my lab, someone in another lab could exactly carry out that. So there are, I think, I agree, it's completely a web that we should all be embracing. And I think that maybe there's a couple of policies that might be suggestive to try and create this reward structure and so that it actually happens by itself.
Like to respond, Glenn?
If I was king of the world, the one thing that I would do would be insist that every research study was performed blinded. So Len mentioned earlier that we, when I was at Amgen, were only able to reproduce about 11% of the studies that we tried to reproduce. What's even more shocking is that on the many, many occasions, we went to the laboratory that had originally reported those results, and they themselves were unable to reproduce their own work in their own laboratory with their own reagents when they were asked to do them with an Amgen scientist standing next to them, and they were performed blinded. So if I could only do one thing, my policy would be every study, I recognise that there'll be challenges, but still every study should be performed blinded. It's much easier to get the result that you want if you know what you're looking at. So, and we all want the result we want. We're human beings. But to try and at least be a little bit more objective, my one policy would be everything has to be performed double blind.
Any other comments? Or?
I think that's a really interesting concept, and what I have experienced in many situations is that when somebody is doing an experiment, they tend to do it a bunch of little things that they don't even recognize have an impact on results. And as soon as you hand a protocol to somebody else and it doesn't have that detail in and they try and do the same thing, they don't get the same results. So I think doing something like that definitely could have an impact.